This is the presentation on the lymphatic system and immunity. This is an area which I've had quite a bit of experience in and have much interest in as well. Okay, so first of all, the lymphatic system is really a collection of cells and biochemicals that travel in your lymphatic vessels. And it's a network of vessels. They help in circulating fluids in your body. They're very closely associated with the cardiovascular system. So they pretty much run somewhat parallel to your blood vessels, except they're kind of siphoning off fluids from your tissues and taking them back to the heart. And at the same time, they have other functions as well. So these three functions, they transport excess interstitial fluid away from the interstitial spaces and they return it to the bloodstream. They absorb lipids from the digestive system and transport them to the bloodstream. And this is accomplished by the lymphatic capillaries that are called lacteals. And also the lymphatic system defends the body against disease and allows humans to live in a world with other organisms. And at this point, we call it the immune system. It has to do with the cells of the lymphatic system that give you defense against disease and also permanent immunity against future infections. And this shows you some of the network that we're talking about and its connection to the cardiovascular system. Here you have the lymphatic capillaries that run pretty much beside your vascular or your blood or cardiovascular capillaries. Okay, and they do have one-way valves very similar to your veins. The fluids are also moved very similarly to the veins, using pressure from the muscles pushing on them. And you have lymph nodes at certain junctions that are going to be important for immunity, where you're going to find certain types of white blood cells. And eventually, the flow of those fluids does end up back in the venous system and back to the heart. Okay, so the lymphatic pathway starts with lymphatic capillaries, then goes to lymphatic vessels, then lymph nodes, and larger lymphatic vessels, lymphatic trunks, lymphatic collecting ducts, and then the subclavian vein in the thorax to go back into circulation. The lymphatic capillaries are microscopic closed-ended tubes. The networks parallel blood capillaries throughout the body. They're thin-walled. The walls are formed from simple squamous epithelium, just like the other capillaries, so they do allow movement of materials between the outside and the inside. The tissue or interstitial fluid enters the lymphatic capillaries, and now we call this fluid lymph. I'm sure you've heard of it referred to that way. Merges into the lymphatic vessels. And this shows those associations at the regular capillaries and your tissues. Okay, the lymphatic vessels. The walls are similar to veins, but they're actually quite thinner. The lymphatic vessels are composed of three layers: an inner layer, which is an endothelial lining, a middle layer, which is smooth muscle and elastic fibers, and then an outer layer, which is connective tissue. And it contains semilunar valves. This allows one-way flow. These are the valves I was referring to a while ago. The larger vessels lead to lymph nodes and then to larger lymphatic trunks. And this is a slide of a lymphatic vessel showing you the one-way valve. You have the lymphatic trunks. They drain the lymph from the lymphatic vessels. They're named for the regions they serve. So you got the lumbar, the intestinal, the intercostal, the bronchomediastinal, the subclavian, and the jugular. And they drain into the lymphatic collecting ducts. The lymphatic collecting ducts drain the lymph from the trunks, and there's two for the entire lymphatic system. you have a thoracic duct. It's longer and wider of the two ducts. It drains the majority of your body. And it begins as a sac that we call the cisternae chyli. It empties into the left subclavian vein. The right subclavian duct is much smaller than the thoracic duct, begins in the left thorax and empties into the right subclavian vein. It drains the upper left portion of your body. Okay, and this shows lymphatic trunks and the collecting ducts. This shows the pathways in the drainage of each of those major ducts. And again, notice left and right drain different areas in terms of proportion of the body. This is a summary of the lymphatic pathway that we had just gone over. Okay, let's talk about the tissue fluid, the lymph. So lymph is tissue fluid that's entered a lymphatic capillary. Lymph formation depends on the tissue fluid formation. And tissue fluid formation includes, so capillary blood pressure filters the water and the small molecules from the plasma. And the resulting tissue fluid has about the same composition as your blood plasma. It has water, dissolved nutrients, gases, and hormones. An exception, tissue fluid does not contain large plasma proteins, which remain in the blood plasma, so that we can maintain osmotic pressure that's necessary to draw fluid back into the blood capillaries. So tissue fluid is the blood plasma minus the plasma proteins. Filtration from the plasma normally exceeds reabsorption, which leads to the net formation of tissue fluid. So this increases the tissue fluid hydrostatic pressure within the interstitial spaces, which forces fluid into the lymphatic capillaries and forms the lymph. This process prevents accumulation of excess tissue fluid, or what we call edema. An example of edema, when a woman has axillary lymph nodes removed during breast cancer surgery. Lymphatic drainage will be obstructed from the upper limb, and this results in edema. Lymph inside the lymphatic vessels has low hydrostatic pressure, like the venous blood. Good lymphatic flow is going to require help from several mechanisms. So muscle activity influences the movement of the lymph to the lymphatic vessels. So contraction of skeletal muscles compresses the lymphatic vessels, which moves the lymph. And as I'd said before, this is kind of similar to venous blood flow. Respiratory processes also create low pressure in the thorax and high pressure in the abdomen during inspiration. And this sends lymph from the abdomen to the thorax. Smooth muscle in the larger lymphatic vessels contracts to aid in flow of lymph as well. And you have valves in the lymphatic vessels that prevent backflow. So it's a one-way valve. So lymphatic flow is the highest during physical exercise. So the roles of the lymphatic capillaries: absorption of dietary fats in the small intestine and delivery to the bloodstream. Return of small proteins that are filtered by the blood capillaries to the bloodstream. Collection of the excess interstitial fluid. Delivery of excess fluid to the bloodstream. Delivery of foreign particles to the lymph nodes. And flap-like valves between the cells of the lymphatic capillaries will allow easy entry of the tissue fluids. Lymphatic tissue contains several cell types, including lymphocytes and macrophages. Mucosa-associated lymphoid tissue, or MALT, we have unencapsulated lymphatic tissue of the digestive, respiratory, urinary, and reproductive tracts. And tonsils and appendix are composed of lymphatic nodules. These are compact masses of lymphatic tissue. And Peyer's patches are aggregates of lymphatic nodules that are found in the ileum, which is the distal part of the small intestine. So all of this is the mucosa-associated lymphoid tissue. The lymphatic organs consist of encapsulated lymphatic tissue and lymph nodes, the thymus, and the spleen. The lymph nodes are usually bean-shaped, and they're less than 2.5 centimeters long. They're located along lymphatic vessels. They filter pathogens from the lymph. They contain lymphocytes to attack viruses, bacteria, and parasitic cells. They contain macrophages to engulf and destroy foreign substances, damaged cells, and cellular debris. Lymph nodes are found in groups or chains along the paths of the larger lymphatic vessels throughout the body. Major locations of the lymphatic nodes include the cervical region, the axillary region, the supratrochlear region, the inguinal region, the pelvic cavity, the abdominal cavity, and the thoracic cavity. They're not found in the central nervous system. And this shows some of those actual lymphatic vessels and a lymph node. This shows an outline of what the medulla or central area of the lymph node would look like, as well as the associated lymph vessels. As you can see, within the medulla, you would find macrophages and specifically T cells or T lymphocytes. Okay, in the germinal centers of the lymph node, you find the B cells. The B cells are what produce antibodies. This picture shows you the possible locations of lymph nodes throughout the body. As you can see, there are key points of where certain pathogens could potentially get into the body. The two primary functions of lymph nodes include filtering potentially harmful particles from the lymph, and then immune surveillance. This is where we monitor the body fluids via macrophages and lymphocytes. Along with the red bone marrow, the lymph nodes are centers for lymphocyte production. Lymphocytes attack various pathogens in the lymph nodes, and macrophages engulf and digest foreign substances, damaged cells, and debris. Okay, one of the clinical applications, lymphedema, you may have heard of. So we have a fluid accumulation in a local area that results from disruption of the flow of lymph. And this often results when lymph nodes are surgically removed because of maybe the presence of cancer. It commonly occurs in cases of breast cancer when cancer is spread to the axillary lymph nodes. So when checking a breast cancer patient for possible spreading of cancer, cancer cells are often found in the sentinel lymph nodes, the nodes in the axillary drainage path from the breast to which cancer cells are most likely to spread first. Lymphedema from breast cancer is found in the arm on the side of the body from which the lymph nodes have been removed. Treatments include regular exercise, wearing a compression sleeve on the affected arm, raising the arm above the level of the heart, and using a compression pump to help drain the excess fluid. Okay, another organ of the lymphatic system, the thymus. It's a soft bilobed gland. It's in your mediastinum. It's divided into lobules. The lobules contain lymphocytes that are derived from progenitor cells and red bone marrow. Most cells are inactive, and we call them thymocytes. Some cells mature into functional T lymphocytes, or what we call T cells, which leave the thymus to provide immunity. Thymosins are hormones produced in the thymus, and they stimulate T cell maturation. They're large in infancy and early childhood, they shrink at puberty, and they're small in the adult. In the elderly, lymphatic tissue is replaced by adipose and other connective tissues. Okay, so this shows you where you would find the thymus. You can see it's right above the heart, over the trachea, really over the aorta. And this shows you size comparisons of the thymus in a fetus versus an adult. Notice it's much larger in the fetus. Okay, the spleen, it's the largest lymphatic organ. It's located in the upper left part of the abdominal cavity. It resembles large lymph nodes. It contains venous sinuses that are filled with blood. It contains two tissue types, the white pulp, which contains lymphocytes and macrophages, and the red pulp, which contains red blood cells and many macrophages. It filters the blood like the lymph nodes filter lymph. It breaks down worn out red blood cells. Macrophages engulf and destroy any foreign particles, and lymphocytes defend the body against infection. Okay, and this shows a picture or drawing basically of the spleen, artistic rendering. And here we actually have a cross section slide of what the spleen looks like. Okay, the major organs of the lymphatic system summary is found right here, this table 16.1. All right, now let's talk about immunity. So the body defenses against infection. The lymphatic system defends the body against infection by pathogens. And pathogens are disease-causing agents like bacteria, viruses, complex microorganisms like protozoa, and spores of organisms like fungi. And these may be single-celled or multi-celled. The presence and multiplication of a pathogen in the body, if unchecked, can cause infection. Viruses have a simpler structure than living cells, and they're not considered to be organisms. Viruses consist of simply a nucleic acid like DNA or RNA and a protein coat. Some examples are herpes viruses, which are DNA viruses, or SARS-CoV-2. This is an RNA virus that causes COVID-19. Viruses use host cells to produce their own proteins and reproduce, and it's one of the reasons why we don't consider viruses an actual organism. Bacteria, they are single, simple cells. They cause many common infections, such as those of Staphylococcus and Streptococcus. So bacteria are very simple types of cells with no nuclei. Okay, types of immune mechanisms. Immunity is the ability of the body to prevent pathogen entry or destroy any pathogens that enter the body. The immune mechanisms include innate or non-specific defenses and adaptive or specific defenses. The innate non-specific defenses are general defenses. They protect against a lot of different types of pathogens. Adaptive specific defenses, they're more specific and precise. They target very specific antigens or specific molecules on those foreign invaders. Adaptive specific defenses are carried out by lymphocytes that recognize certain foreign molecules. So here we can start referring to T and B lymphocytes now with the adaptive immunity. Okay, innate defenses, species resistance and mechanical barriers. In species resistance, certain species are resistant to diseases that affect other species. Certain species lack receptors, temperature, or chemical environments for a particular pathogen. So this is one reason why some organisms can't get diseases from us, and we may not be able to get certain diseases from other organisms. Mechanical barriers. This includes skin and mucous membranes. It prevents the entrance of the pathogen at a kind of a simplistic level. It's considered the first line of defense. All other non-specific defenses are part of the second line of defense. So examples, epidermis sloughs off. So you just remove the upper part of your skin regularly, and that will help remove superficial bacteria. Ciliated epithelium in your respiratory tract. This traps and sweeps away pathogens. Hair traps pathogens. And tear, saliva, and urine wash away microorganisms. Again, these are non-specific mechanical barriers. Okay, inflammation. Inflammation produces local redness, swelling, heat, and pain. Redness is due to vasodilation, so your blood vessels dilating or swelling. Swelling results from increased capillary permeability and fluid entry into the tissue spaces. Heat is derived from blood arriving from the deep areas of the body. Pain is due to stimulation of pain receptors. A process that walls off infection site and inhibits the spread of infection, in this case, and white blood cells gather in certain areas and destroy pathogens by phagocytosis. Exudates are fluids that contain fibrinogen, and other clotting factors may form fibrin networks. Following control of infection, the phagocytes engulf and destroy the dead cells, and then cells that were lost will be replaced by cell division. And this shows you the major actions of an inflammation response. Chemical barriers. These include enzymes in the body fluids that provide a chemical barrier to pathogens. Some examples include pepsin in the gastric juice and lysozyme in your tears, which destroy microorganisms. Interferons, they block viral replication. They act against the growth of tumors, and they stimulate phagocytosis. Defensins, these are peptides produced by neutrophils and other granulocytes. They cripple microbes by making openings in the cell membranes or the walls. Collectins, these are proteins that protect against many bacteria, yeast, and some viruses. And then there is the complement system, which is a group of inactive proteins in the plasma and other body fluids that become activated by binding to the surface of foreign cells. Once they're activated, it begins a cascade of reactions that stimulate inflammation, attracts phagocytes, enhances phagocytosis, and causes cell lysis. Okay, the innate defenses include natural killer cells or NK cells. This is a small population of lymphocytes. They're very different from the B cells and T cells that provide adaptive defenses. They defend against viruses and cancer cells by secreting cytolytic substances called perforins that lyse the cell membrane. They enhance inflammation. Phagocytosis, this is also an innate defense. It removes foreign particles from the lymph. Phagocytes in the blood vessels and the tissues of the spleen, liver, or bone marrow will remove particles from the blood. Most active phagocytic cells are the neutrophils and the monocytes. Chemicals from damaged tissue attract these phagocytic cells to the injury. This is called chemotaxis. Monocytes that leave the blood become macrophages, which can be free or fixed in tissue. And mononuclear phagocytic systems, or reticuloendothelium. This consists of the monocytes and macrophages of the body. Okay, another innate defense, fever. This begins when a viral or bacterial infection stimulates the lymphocytes to proliferate. And this produces cells that secrete a substance called interleukin-1, which raises thermoregulatory set points. Interleukin-1 is also called endogenous pyrogen, or firemaker from within. Elevated body temperatures indirectly inhibit microbial growth and causes liver and spleen to take up iron, making it unavailable for the bacteria and the fungi to use in their normal metabolism. High body temperature also increases phagocytic activity. Okay, table 16.3 here goes over the different types of innate or non-specific defenses, which we talked about. Okay, adaptive specific defenses. So characteristics of adaptive defenses include, well, for one thing, they're called the third line of defense. Okay, that's resistance to particular pathogens or to their toxins or metabolic byproducts. It's based on the ability to distinguish molecules that are part of the body, which we call self, from those that are not, which are non-self. Antigens are non-self molecules that can evoke an immune response. So an antigen is a marker or a molecule on that other organism or that piece of material. Adaptive responses are carried out by lymphocytes and macrophages that can recognize specific antigens. And there's two types of adaptive defenses: cellular immune response, which is performed by immune cells, and humoral immune response, which is performed by the antibodies. So overall, kind of summarizing up to this point, pathogens begin to enter the body, and the first line of defense takes care of a lot of it. These are the mechanical barriers like skin and mucous membranes. Then pathogens enter the body. And the second line of defense gets involved with chemical barriers like enzymes, pH, salt, interferons, defensins, collectins, and complement, or natural killer cells, or inflammation, or phagocytosis, or fever. Okay, and then the third line of defense gets involved at the cellular immune response level or humoral immune response level. Let's talk more about antigens. These are basically receptors on the surface of the lymphocytes. Oh, let me try this again. Receptors on the surface of lymphocytes will enable the cells to recognize the non-self antigens. And again, those antigens are just like molecules that are recognized on the surface of those other things as being non-self. The lymphatic system responds to non-self antigens, but not your self antigens under normal circumstances. Of course, that's a very good thing. You don't want your body acting against your own materials. If you do, then you start destroying your own body, and that's what happens in autoimmune diseases. Antigens can be proteins or polysaccharides or glycoproteins or glycolipids. The most effective antigens are large and complex molecules. Haptens are small molecules that are not antigenic by themselves, but when they combine with a larger molecule in the body, then they can evoke an immune response. Okay, lymphocytes are produced throughout life, starting during the fetal development. Red bone marrow releases unspecialized lymphocyte precursors into the blood, and half of these cells will settle in the thymus and specialize, and then they will be released. These are the ones that will become the T cells, and it makes sense since they're made, basically, or they mature, I should say, in the thymus. The other half of the cells differentiate in the red bone marrow and are then released, and they become B cells. Now, B cells are not named for bone marrow, although you can use it to help remind yourself of it. They were actually named for a small organ they were found in birds originally, called the bursa fabricius. The main types of lymphocytes include T lymphocytes and B lymphocytes. The T lymphocytes are also called T cells or thymus-derived lymphocytes. They specialize in the thymus. After release from the thymus, they make up 70 to 80% of your circulating lymphocytes. Some T cells will settle into lymphatic organs, such as lymph nodes, thoracic duct, or the white pulp of the spleen. The B cells, the B lymphocytes, also called the B cells. Again, B stands for bursa fabricius, from specific bird types of organisms like chickens. And after the release from bone marrow, they make up 20 to 30% of lymphocytes in blood. They're abundant in lymph nodes, the spleen, the bone marrow, and intestinal linings. And this is a good image to help you remember where those types of lymphocytes are produced and where they mature or specialize. Table 16.4 shows you a comparison of the T cells and B cells. And to go over the primary functions here, since we haven't talked a lot about that yet, primary functions of the T cells are to provide cellular immune response where the T cells interact directly with the antigens or antigen-bearing agents to destroy them. The B cells provide humoral immune response in which the B cells interact indirectly by producing antibodies that destroy the antigens or antigen-bearing agents. So the B cells produce antibodies. Let's talk more detail about the T cells. A lymphocyte has to be activated before it can respond to the presence of an antigen. T cell activation requires that antigenic fragments be attached to antigen-presenting cells, or an APC. The APC basically phagocytizes the antigen. So it basically ingests it, then digests it. Then it displays the antigenic fragments on its cell membrane, bound to a protein that is part of what we call the major histocompatibility complex, or MHC, or also called the human leukocyte antigen, HLA. When specific T cells encounter an APC displaying foreign antigenic fragments bound to the MHC proteins, it can now recognize the antigens as foreign. Now the T cell is activated. and the T cells can secrete polypeptides called cytokines, which enhance some cellular response to the antigens. So this is another function of cells that ingest or phagocytize. They take in that organism, and yes, they help destroy it, but they also basically alert the T cells to these foreign particles or fragments or organisms. Types of specialized T cells include the helper T cells, which activate other cells by secreting cytokines. They stimulate B cells to produce antibodies, and they stimulate the activity of cytotoxic T cells. Cytotoxic T cells attack virally infected or cancer cells. Memory T cells provide future immune protection. And regulatory T cells suppress immune responses after defeat of pathogens, which lowers the chance of developing an autoimmune disease. There's two types of MHC proteins, which act as antigen-presenting cells for certain types of T cells. Okay, the class 1 MHC proteins, they're found on the surface of all your nucleated cells. They form complexes with the self or non-self antigens that's entered the cells and display them on the surface of the cell. Cytotoxic T cells recognize and bind to the class 1 MHC protein or antigen complex. Self-antigens do not evoke an immune response from the cytotoxic T cells. The foreign or non-self antigens do activate cytotoxic T cells and evoke an immune response from them, called the cellular immune response or cell-mediated immunity. Okay, class 2 MHC proteins. These are found on the surface of certain cell types like macrophages and B cells. Only these cell types can act as antigen-presenting cells for the helper T cells. The antigen-presenting cells display the antigen class 2 MHC protein complexes on their own surfaces or cell membranes. Once a helper T cell recognizes its displayed foreign or non-self antigen, it is now activated. Activated helper T cells can now begin the cellular immune response or cell-mediated immunity. Helper T cells now secrete a variety of cytokines, which enhance certain cellular responses to antigens. Okay, speaking of cytokines, there are several different types, like colony-stimulating factors that stimulate bone marrow to produce lymphocytes, or interferons that block viral replication and stimulate macrophages to engulf viruses, and stimulate B cells to produce antibodies and attack cancer cells, interleukins which control lymphocyte differentiation and proliferation, and tumor necrosis factor which stops tumor growth, releases growth factors, causes fever that accompanies bacterial infection, and stimulates lymphocyte differentiation. Okay, let's talk more about the B cells and humoral immune response. So B cells can be activated when an antigen fits the shape of their receptors and binds to them. Further B cell activation requires cytokines from T cells. Once proper cytokines are released from helper T cells, B cells respond by proliferating and enlarging the clone of their identical cells. Some new B cells become memory B cells, which provide future immunity. Other new B cells differentiate into plasma cells, which produce and secrete large globular proteins called antibodies, or also called immunoglobulins. Since antibodies are carried by the blood, which is a body fluid, to the infection site, this type of response is called the humoral immune response, or the antibody-mediated immune response. Okay, figure 16.8 is a good graphic that helps you understand how the B cells respond, as well as how antigen-presenting cells work, and how helper T cells are influenced, and how they influence other cells to ultimately activate memory T cells and cytotoxic T cells. Okay, figure 16.19 shows B cell proliferation. Okay, and notice the antibodies being produced by the B cells ultimately. Okay, those antibodies are originally just receptor markers on the cells, and then they get released, and they are basically the antibody ultimately. Okay, table 16.6 goes through the steps in antibody production, okay, which we had gone over already, but this summarizes all of those activities. Okay, what are antibodies? Well, they're globular proteins. They make up the gamma globulin fraction of plasma proteins. They're also called immunoglobulins. They're Y-shaped proteins composed of 4 amino acid chains, 2 heavy chains and 2 light chains. Each type of antibody has a unique amino acid sequence and conformation, making it specific for its antigen. The unique site in the variable region in which the antibody binds to its antigen is called the antigen binding site. The other portions are called the constant regions because they are similar to those of the other antibodies. There's 5 major types of antibodies, or immunoglobulins. IgG, this is 80% of the antibodies. They act on bacteria, viruses, and toxins. IgA, this is 13% of antibodies. They're found in exocrine gland secretions. IgM, this is 6% of antibodies. They act on antigens in foods and bacteria. IgD, this is less than 1% of the antibodies. They're found on B cell surfaces and are common in infants. And IgE, which is less than 1% of antibodies and found in exocrine gland secretions. Figure 16.20 shows you the antibody molecule. Okay, at the top, at the Y, is where we have the antigen binding sites. This bottom part is the constant region. These light blue are the variable regions. They're the ones that bind to different types of antigens. Okay, table 16.7 goes over the characteristics of the major immunoglobulins. By the way, you may have heard of gamma globulin before. That's the same thing as IgG. Okay, what are the actual actions of the antibodies? Well, the antibodies respond to antigens in 3 different ways. The methods of direct attack include agglutination, precipitation, and neutralization. In agglutination, you have clumping of the antigens. This makes phagocytosis easier by the phagocytic cells that engulf them. Precipitation, this makes the antigens insoluble. Again, this makes phagocytosis easier. And then neutralization. It covers the toxic portions of antigens, which make them harmless. You can also have activation of complement. This is more effective against antigens than the direct attacks. It occurs with certain IgG and IgM antibodies. Actions of the activated complement proteins include agglutination, which clumps the antigen-bearing cells, opsonization, where you coat the antigen antibody complexes, chemotaxis, which attracts macrophages and neutrophils, lysis, which ruptures the cell membranes of the pathogens, and neutralization, which changes the structure of viruses so they're harmless. Stimulation of local change inflammation to help prevent spread of infection also occurs. IgE antibodies on mast cells bind to the antigens, and antigens produce histamine. Inflammatory chemicals will cause edema and vasodilation. Figure 16.21 goes through the different actions of the antibodies against antigens in a pictorial way, which may be helpful to you. Table 16.8 goes through the actions of antibodies in summary, as we have discussed. Okay, let's talk a little bit of technology and how we make use of the immune system. And immunotherapy. Immunotherapy makes use of the immune system components to fight disease. The humoral immune response by antibodies and the cellular immune response by cytokines. Monoclonal antibodies. A specific B cell which produces a single type of antibody, or monoclonal, can be fused with a cancerous cell to produce a cell that produces one type of antibody, and it keeps dividing. These can be used in research, agriculture, pregnancy tests, detecting new or recurring cancers, delivering medications specifically to tumors, early therapy for mild to moderate SARS-CoV-2. Then we have cytokines that can be used. They can now be produced in large quantities because of recombinant DNA and monoclonal antibody techniques. The cytokines are used to treat leukemia, multiple sclerosis, hepatitis, genital warts, melanoma, and kidney cancer. And then CAR T, or chimeric antigen receptor T cell. This therapy uses the body's immune system to detect and destroy specific cancer cells. DNA instructions are used to genetically modify a T cell protein, which is part T cell receptor and part monoclonal antibody against a tumor antigen. After culturing these modified T cells, they are infused into the patient, where they bind to the specific tumor cells and alert the immune system to attack those specific cells. Okay, let's talk about immune responses. So primary immune responses are produced by the first encounter with the antigen. The B and T cells specific for the antigen become activated. Plasma cells then release IgM and then IgG antibodies. So the IgM are the first ones that are produced. The first antibodies appear in 5 to 10 days, and they remain for several weeks. Memory B cells are then produced also. In secondary immune response, this is a response to subsequent exposure to the same antigen. It produces high concentrations of antibodies in 1 to 2 days. It's accomplished by the memory B cells and memory T cells. And these antibodies remain for months or years. Memory B cells can live for many years. Okay, so secondary immune response is immunity. Okay, and it's one reason why you normally don't even know you've been infected a second time, because it acts so quickly. But again, it depends on the particular antigen, and it depends on if that antigen is able to mutate quickly. Okay, figure 16.22 shows you the difference in the primary response versus secondary. Notice in terms of days, the secondary immune response happens much quicker. And notice in terms of antibody concentration, it's a much higher concentration of antibodies as well. Okay, practical classification of immunity. Classification of immunity is based on how it's acquired. Naturally acquired is obtained by a natural process, such as getting or recovering from the disease, or given from mother to the fetus or infant. Artificially acquired means obtained by an injection instead of natural processes. Also, classification of immunity can be based on whether it's temporary or permanent. In passive immunity, we have temporary immunity that's obtained via antibodies. In this case, no antigen exposure has occurred. No immune response is evoked in person's immune system in passive immunity. It just means you're getting the antibodies somewhere else. You're not producing them. Active immunity is permanent immunity, and that is gained by getting the antigen, and your own system is evoked, and you produce the antibodies, and you produce memory B cells. And we can combine all of these terms in different ways, such as naturally acquired active is an exposure to a live pathogen. And the result is stimulation of an immune response with the symptoms of disease. So that means you actually got the disease. You got infected naturally from your environment. Artificially acquired active means you're exposed to a vaccine quite often. So either you're getting a weakened form of that pathogen or a dead pathogen or components of the pathogen in your vaccine. Those are all different ways we can create vaccines. So your immune system is still stimulated. So it's just like you had had the disease, except you don't get the actual disease. You just get the organism parts. Okay, naturally acquired passive immunity. Antibodies are passed to the fetus from pregnant woman with active immunity or to the newborn through colostrum or breast milk from a woman with active immunity. Okay, again, it's passive, so you do not form memory to it. Short-term immunity for a newborn without stimulating an immune response. And then you have artificially acquired passive immunity. In this case, you are given an injection, but you're given an injection of the actual antibodies. We can call this anti-serum. We can also call this antitoxin. It gives short-term immunity without stimulating an immune response. Again, it does not give you long-term immunity. You do not form memory cells. Okay, there are different types of hypersensitivity reactions evoked by the antibodies. A hypersensitivity is an exaggerated immune response to a non-harmful antigen. The person is pre-sensitized to a certain antigen. Some can affect almost anyone. Some can affect only people with inherited ability to have an exaggerated immune response. And there's 4 types of hypersensitivities. Type 1 is immediate reaction hypersensitivity. This is what we call an allergy. The person produces a lot of IgE antibodies against a specific allergen. This occurs in minutes after contact with the allergen, and it evokes histamines to be released. And this may cause hives, hay fever, asthma, eczema, gastric disturbances, anaphylactic shock, which is a life-threatening form. Type 2 hypersensitivity. This is antibody-dependent cytotoxic reaction. The antigen binds to a specific cell and causes phagocytosis and complement lysis antigen. Transfusion reaction is a type 2 reaction. Okay, type 3 hypersensitivity or immune complex reactions. Antigen-antibody complexes form and deposit in certain tissues in this case. It damages tissue via phagocytosis and complement binding. Antibody complexes cannot be cleared from the body in this case. This is like rheumatoid arthritis. Type 4 or delayed reaction hypersensitivity. This can affect anyone. It results from repeated exposure of skin to allergens. The allergen activates T cells in skin, which release chemicals that cause eruptions and inflammation. And it takes about 48 hours to occur. Okay, table 16.10 goes over those hypersensitivity reactions. Okay, 16.23a is a figure that shows immediate reaction allergy and the steps. Okay, transplantation and tissue rejection. This happens because of your immune system. Tissues and organs that can be transplanted include bone, cornea, kidney, liver, pancreas, heart, bone marrow, skin, and lungs. When the donor's tissues are recognized as foreign, there's a tissue rejection reaction. The recipient's immune system will try to destroy the foreign tissue. In graft versus host disease, we have damage to the recipient's tissues by molecules in the immunologically active transplanted tissue, like bone marrow. In rejection reactions, this resembles the cellular immune response against antigens. It's important to match the MHC antigens between the donor and the recipient. The greater the antigenic difference, the stronger and faster the rejection reaction will occur. Immunosuppressive drugs can be used to prevent rejection. Well-matched transplants may not require lifelong immunosuppressive drugs if bone marrow stem cells from the donor are given along with the transplant. Types of grafts and tendency for rejection. In autografts, this means you actually get a graft from another part of your body. So that's autograft. Isograft means you get it from an identical twin. So these would not cause rejection reactions because it thinks it's your body. in allografts. This is from one person other than the identical twin. Xenografts are from different species. These do tend to cause rejection reactions. Okay, table 16.11 goes through these transplant types. Okay, in autoimmunity, this is an attack by the immune system against your own tissue. The immune system fails to distinguish itself from non-self, and the body produces antibodies called autoantibodies. Cytotoxic T cells also attack the body's tissues and organs. Various autoimmune disorders affect different types of cells. There are several theories concerning the causes of autoimmune disorders, but no single one cause has been established. Table 16.12 goes over several types of autoimmune disorders that you may want to pause this video or run through when you have time. Okay, clinical applications, immunity breakdown, and HIV/AIDS. So HIV is human immunodeficiency virus. It is a virus that breaks down the immune system function. So this virus, one of the reasons why it was so dangerous to start with was because it attacked the very system that's supposed to prevent infection. It may stay silent for years and then progress to AIDS or acquired immune deficiency syndrome. Then an opportunistic infection begins. HIV attacks the macrophages and then the helper T cells. When the helper T cell numbers decline, B cells cannot produce antibodies due to lack of cytokine activation. Later, HIV variants affect cytotoxic T cells too. The person dies from loss of immune response against pathogens and gets cancers. Modes of transmission include sexual contact, contaminated needles, birth or milk from infected mother, receiving infected blood or tissues from a donor. Okay, your lifespan changes for the lymphatic and immune system. Your immune system function declines early in life as the thymus gland shrinks. Only 25% as powerful as it once was once you're an adult. The strength of the immune response decreases in elderly people. There's a higher risk of infection, cancer, diseases like pneumonia as a person ages. This is one reason why you see cancers in more elderly, older individuals. T cell numbers decrease very slightly. B cell numbers do not change, but activity levels decline in both types of lymphocytes. Antibody response to antigens becomes slower. IgA and IgG antibodies increase. IgM and IgE antibodies decrease. Vaccines may require extra doses. More autoantibodies may be produced, increasing the chance of developing an autoimmune disease. And the elderly may not be candidates for certain medical treatments that suppress immunity, such as chemotherapy. And that is the end of this section on the lymphatic system and the immune system.